Moving on, we'll hear the presentation of Dr. James Leong from Arizona State University on something that's more in the transition between geology and biology, I guess. No, James?
Yes.
Okay, take it away.
Sorry, slideshow. Okay, so I'll just do the presentation mode, and okay, I'll just hide our faces here. Okay, so hello everyone. Magandang umaga sa inyong lahat. I'm James Leong, a graduate of UP-NIG some years ago, and recently from Arizona State University. And sa sunod na mga minuto, I will talk about my PhD research at ASU with my advisor, Professor Everett Shaw. So my PhD is mostly aqueous geochemistry, mineralogy, and geochemical modeling. But however, today I'll talk more about the connection between a part of my research, deciphering or looking into the connection between the geosphere and the biosphere, and how rock composition dictates fluid composition and ultimately dictates how microbes can take advantage of the chemicals that are present in these fluids that are resulting from the water-rock interaction. So let's start. So the geosphere is directly involved in the habitability of our planet. Interaction between water and rocks mobilizes nutrients and facilitates the transfer of energy from the lithosphere and the biosphere. One remarkable process is when fluids coming from the deep subsurface discharge back into the surface, like in hot springs, if you're in the continent, or if you're in the seafloor, hydrothermal vents. And there's also interesting low temperature features like very, very alkaline springs that have reacted with the rock that is also supporting a lot of microbes. Not a lot of microbes, but interesting microbes. So despite the extreme conditions in this environment, high pH, high temperature, very, very enriched in certain chemicals, we know that life thrives in this environment. Interestingly, especially those at the deep seafloor, where the energy from the sun cannot penetrate, life is primarily sustained by chemical energy. So the base of the food chain in these ecosystems are microorganisms known as chemotropes or chemosynthetic microbes, which are basically fueled by chemicals venting out from these deep-seated fluids. So the alteration of different rock types can lead into the formation of hydrothermal and low temperature fluids and volatiles that are different in compositions. So different rocks will react differently and will result in different fluids. And I hope by the end of this talk, I will be able to impart that these differences in the composition of fluids and volatiles can lead to varying potentials to support different microorganisms. So hydrothermal vents are a spectacular consequence of water-rock interaction. This can happen at temperatures exceeding 300 degrees Celsius, as exemplified by black smoke systems like in Logochev near the Mid-Atlantic Ridge, venting very hydrogen and methane-rich fluids. And this process can also occur at low temperatures down to 100 degrees Celsius, such as those occurring at the Lost City hydrothermal vent, Lost City hydrothermal field, also near the Mid-Atlantic Ridge. The scale of the chimneys here are order of meters. So basically, for instance, in Lost City, some of the highest chimneys can reach a height of 60 meters. So that's basically around the 10th, 12th floor building. So even more profound, as shown in the pictures, some remarkable water rock interaction can happen at low temperatures present in certain continental aquifers, and it does not require magmatic heat to proceed. So this type of fluid will be the focus of this presentation. Shown here in these pictures are less than 40 degrees Celsius, even less than 20 or 30 degrees Celsius, fluids seeping out from rocky outputs in the Sultanate of Oman. And from these pictures, you can see that these are not ordinary fluids. These fluids have very high pH, often greater than 11. So that's basically almost equivalent to the bleach solution that you can find in your households. The process that generates this remarkable fluid happens when water reacts with peridotite. So peridotite is a rock type derived from the mantle. Basically, most of the Earth's upper mantle is made up of peridotite. So when these rocks are brought to the Earth's surface through tectonic events, the mantle-derived minerals within this rock are very unstable and will alter into mostly a mineral called serpentine, and hence the term serpentinization to describe this water rock or alteration process. So during serpentinization, a starting fluid with a near neutral pH like seawater or slightly acidic pH like rain becomes very alkaline as it reacts with more and more peridotite until pH attains very high values, like often greater than 11, sometimes reaching pH greater than 13. Aside from being alkaline, these fluids are also very reduced and are enriched in hydrogen and methane gases. So in some places in Oman, you can see that there's very vigorous bubbling over here that you might think that it's boiling, but the temperature is just around 32 to 40 degrees Celsius. So just a very, very low temperature fluids. And you can see that the water here are not ordinary. There's a very, very interesting fluids. So the hydrogen and methane-rich nature of this fluid that is generated during water interactions have profound implications in the habitability of rock-hosted environments. These volatiles, hydrogen and methane, are food source for a lot of microbes. Think of this as like fuel source. That's why we need to eat to fuel our serve as our fuel. Just like these microbes are eating this hydrogen and methane as their fuel source. So overall, mineral-rich hydrothermal fluids and their low-temperature analogs are attractive model ecosystems for assessing the link between the geosphere and the biosphere, as it can enable habitable environments here on Earth, and perhaps water rock interactions can also enable and support life in places beyond our own planet, like in ice-covered ocean worlds in the outer solar system, like Enceladus in this picture. Ocean worlds are prime targets for locating life beyond Earth, as you have the right ingredients. So beneath this thick ice shelves are liquid water in contact with silicate rocks, and water rock interaction, just like here on Earth, can mobilize nutrients and transfer energy between the lithosphere and biosphere. And like here on Earth, this process supporting life, perhaps in this places, it can also enable habitable systems. So basically, water rock interactions, again, mobilizes nutrients and set the stage for the chemical disequilibrium that can power life. Although these ocean worlds represent the frontiers of our understanding between the link between the geosphere and life, we're only starting to accumulate data on the compositions of fluids and volatiles forming in this extraterrestrial environment. Predictions in the possible ranges and compositions of fluids, volatiles, and microbes that might occur in this not so accessible environment can be done through thermodynamic simulations. These simulations can potentially inform what to expect as we are gearing up for future missions into ocean worlds, such as the Europa Clipper, which is basically launching in a few years. That will sniff volatiles from plumes that vent out from the icy shelves of Europa. Europa is a moon of Jupiter. And basically the spacecraft will sniff volatiles and dust that is being vented out in this from this icy shelves, and basically do some chemical analysis. There's a lot of a suite of mass spectrometers within this aircraft that is currently being built right now. And in other words, we are going to do a lot of geology and biology. So basically, we're going to infer a lot of the geology and biology of the interior of this icy moon based on aqueous fluids and volatile data. So to reiterate, thermodynamic models can help us inform what would be this possible numerous compositions of fluids and volatiles in this environment, and perhaps predict what kind of microorganisms can thrive within these ocean worlds. And thus, it is very important that we test these models with analog environments here on Earth. So basically, two examples of microbial reactions that I will talk about in this presentation are methanogenesis and hydrogen oxidation. So methanogens are microbes that produces methane from CO2, while hydrogen oxidizers are microbes that provide their energy source through the oxidation of hydrogen gas. So note that both reactions needed hydrogen. So basically, methanogens and hydrogen oxidizers are all called hydrogenotrophs. So let us look into how water rock interactions generate hydrogen that can basically be ultimately fuel microbes. So hydrogen is produced during the oxidation of ferrous iron within the primary minerals or the original igneous mineral. And during alteration, this ferrous iron will be oxidized to ferric iron in secondary minerals or alteration minerals. Basically, the oxidation of iron is coupled to the reduction of water to hydrogen. So when you oxidize an element, you need to reduce something else. That is, the electron released during the oxidation reaction should find a new home. And in this case, basically water is basically accepting that electron and basically reduced to hydrogen. The alteration of different rocks will lead into different secondary minerals that controls how iron is distributed and speciated. And this in turn dictates how much of the starting iron can be oxidized and hence the amount of hydrogen produced. Ultimately, water reacting with different rock types or different kinds of rocks will result in fluids with different aqueous and gaseous composition, and as well as concentration. In particular, different amounts of hydrogen that is being produced. So shown here in the diagram is an example of that variations. So this diagram plots the hydrogen concentration in log molality in the y-axis of numerous hydrothermal, high temperature, and low temperature fluids sampled in the seafloor as well as in continents in two different rock types. The greens are fluids sampled in peridotite bodies, while the oranges are mostly sampled from basalt-hosted systems, with a few from gabbros here in Oman. So peridotites, just a little bit background petrology, are magnesium-rich rocks that makes up most of the upper mantle. Basalts and gabbros are rocks that form from the magma generated during the melting of mantle peridotites. So hence gabbros and basalts are compositionally similar. So gabbros are more, the difference is just gabbros are texturally more coarse-grained as they solidified at depth, whereas basalts are fine-grained because they are extrusive rock. They are formed from the rapid solidification of magmas at the surface or near the surface. So between these two types of rocks, compared to peridotites, gabbros and basalts are less magnesium-rich but are more silica and aluminum-rich. So note that we typically find the most hydrogen-rich fluids in peridotite-hosted systems. The difference is not that much at high temperature systems, but as you go to lower temperature systems, the difference can be orders of magnitude. So thus we see significant hydrogen production during water rock interactions with peridotites relative to basalts and gabbros. So now that we have a glimpse of water interacting with different rock types yields different gas compositions or fluid compositions that may be used by microbes, we can predict that these two different fluids can have varying potentials to fuel or to support different ecosystems. So how do we quantify energy available to microbial metabolism? So chemolithic life or chemosynthetic life requires the presence of chemical species that are out of equilibrium with each other. So like hydrogen in the presence of oxygen or hydrogen in the presence of CO2. We can quantify how far is this from equilibrium or how much is the disequilibrium using a term called affinity, represented by the letter A here. Affinity is just a negative of the total Gibbs free energy of the reaction. And the total Gibbs free energy of the reaction is basically just the difference in the ratios of the product and the reactants of a reaction measured in a given system, represented by Q, or the reaction quotient, to what this ratio should be if it's in equilibrium, it's represented by K, or the equilibrium constant. So K is something that we can calculate using a thermodynamic calculator of standard state Gibbs free energy, which is, you can find them online. Whereas Q is something that that can represent ratios in some environmental sample. So thus we need to go to the field and sample this fluid and measure these reactants and products and basically measure and ultimately measure how far are these ratios should compare to equilibrium. So the unit for affinity is basically in calories or joules per mole reactant. And if you have constraints on the limiting reactants of certain microbial reactions, that's in MR in moles per kilogram or M over R here, and multiplying this two, we can get a unit in calories per kilogram, which is an energy unit that is normalized per kilogram of fluid, which is much more tangible than a calories per mole. So I'll be using a calories per kilogram unit in this talk. So for example, let us look at the energy supply from methanogens supported through this reaction. So in order to quantify energy, we need to know Q, or the activity ion product, or the reaction quotient, which is the ratio of activities of the reactants and products of this reaction. So thus we need to go to the field and measure dissolved inorganic carbon, dissolved hydrogen, dissolved methane. And the activity of water is related to the ionic strength. So we need to measure everything, basically all the solutes, so that we can calculate ionic strength. So basically, once we measure almost everything in the dissolved component in the fluids, we can basically calculate Q and hence energy. So we often go to the field where we can get samples. So one place where we can relatively sample this fluid easily as we are on land is in Oman. So here in Oman lies what was once the seafloor. The oceanic lithosphere was placed into the Arabian subcontinent millions of years ago through tectonic events, exposing peridotites, gabbros, and basalts to the surface. And this is a picture of mountains consisting of peridotite rocks in a body called the Samail ophiolite in Oman. Here's a much closer view of the Samail ophiolite, which is just southeast from Dubai. Zooming in, you can see that the rock that makes up the mountains can be distinguished into two different rock types. Basically, the darker ones are peridotites and the lighter ones are gabbros. So we sampled around 140 fluids from a number of places in Oman, accounting for the local host rock type, to be able to see how lithology can dictate fluid compositions and eventually predict which microbes are favored to thrive in the systems in this environment. So just a side note, aside from Oman, mantle-derived peridotites, as well as other components of the oceanic lithosphere, are tectonically exposed in a number of locations here in the Philippines, as shown in this map. The black blocks are depicting different ophiolites here in the Philippines, and hyperaqueous fluids has been documented in the Zambales and Palawan ophiolites. So moving back to Oman, shown here are results of analytical work focusing on just high pH samples or the hyperaqueous samples. And not surprisingly, since gabbroic rocks are more enriched in silica and aluminum, fluids associated with gabbroic bodies are much more enriched in dissolved silica and dissolved aluminum than those hosted in peridotites. So basically the orange are gabbro-hosted fluids, and the greens are the data from peridotite-hosted fluids. Basically just plotting the circle are plotting the average concentration in the fluid, while the bars represent the 25th to the 75th percentile of the fluid samples. So gabbro-hosted fluids, however, have much more lower dissolved hydrogen. The diamond depicts individual samples. Unfortunately, we don't have a lot of samples from gabbro-hosted fluids, so we're just plotting them individually. Unlike for peridotite-hosted bodies, where have 40 or 50 samples. On the other hand, gabbro-hosted fluids have much higher dissolved methane. Gabbro-hosted fluids are also typically enriched in dissolved sulfate and sulfide concentrations. So hydrogen, methane, sulfate, and sulfide are commonly used by many microbes as their energy source. The difference in the concentration of this dissolved species could constrain the differences in their microbial, in which microbial metabolic process is favored to thrive in a given rock-hosted system. And we can calculate this using the equation that I showed earlier. So, and predict which microbe is much more favored in either a gabbro-hosted system or a peridotite-hosted system. And in the following plots, I will plot the calculated energy supplies in calories per kilogram in log form using the equation that I showed earlier. And this is the result for the amount of energy in log calories per kilogram of fluid available to microbes that uses hydrogen in peridotite-hosted systems. So these are all hydrogen neutrophs, hydrogen oxidizer, methanogens, and sulfate reducers. They are all using hydrogen. And you can a value of 0 here means that you have 1 calorie available per kilogram of water to a population of hydrogen oxidizers. A value of -2 means 10 microcalories per kilogram of fluid. And this is how energy supply trends with gabbro-hosted fluids. And note that energy available for hydrogen and sulfate reactions for hydrogen oxidizer, methanogenesis, and sulfate reducers are much lower than those that can be found in peridotite-hosted systems. And it's obvious because, as I showed earlier, there's not much hydrogen in gabbro-hosted systems, so there's much less energy available for microbes that uses hydrogen. However, sulfate reduction with methane as your electron donor is much more favored in gabbro-hosted systems because gabbro-hosted fluids are much more enriched in sulfate and methane. So microbes that live off using these two chemicals are very much happy, and basically they are predicted to thrive much better in the systems than in peridotite-hosted ecosystems. So overall, this calculation shows us that we can expect what type of microbes that can live in these different rock-hosted systems. Several work in our research group are aiming to test this calculation results with actual analysis of microbes, with community analysis of samples that we brought from the field. And basically, as a summary, we learned that water-rock interactions can facilitate the transfer of energy between the lithosphere and the biosphere, and aqueous alteration or alteration of different rock types can yield fluids of different compositions that can result in different potentials to support different microbes. And the compositions of rocky bodies beyond our planet could be very diverse, and it could be daunting to predict what could be out there. So however, our planet Earth has a rich number of analogs hosted in various ultramafic rock types where we could start studying the systems. And it's very important that we use this Earth analogs to understand the link between host rock compositions and the life it can support to prepare us for upcoming missions into these frontier worlds. So that's it. Thank you.
All right, thanks, James. Ang daming information on, but what I got is for anaerobic microbes, they get more energy in ultramafic rocks, and for aerobic microbes, they get more energy in more silicic rocks like gabbro. Is that correct?
No, basically, the only aerobic reaction that I studied here is hydrogen oxidation. The rest are all
Hydrogen.
anaerobic. So
Okay.
it's not, yes, mostly depending on which gas is much more available in gabbro or in peridotite hosted systems.
Okay.
So, and basically, I didn't talk about this, but in my research, I basically talk about, basically deals a lot with why these different rock hosted systems lead to different fluid compositions, why you have much more hydrogen in peridotite hosted system, while you have more sulfide or sulfate in gabbro hosted system. But unfortunately, I basically just try to focus more on its implications to microbial. What is its implications to what could be possible? What type of microbes could thrive in the systems? But yes, it's much more based on the fluid composition, overall fluid composition.
And the other thing, sorry, I'll ask my question now. Baka makalimutan ko. The other thing is, did your group or any other group try to calculate how much water-rock interaction is happening in the ocean floor, considering that gabbros and peridotites, which are abundant in the ocean floor, actually result to serpentinization and higher pH. So we know that the pH of seawater is buffered, with carbon dioxide, but I don't know what's the effect, or how much the effect is with that equilibrium of water-rock interaction. Just a quick question.
That could be interesting because much of the pH of the seawater right now is basically dictated by atmospheric concentration,
right.
also the flux from rivers and all those things. I don't know how much the flux of seafloor derived water rock interaction is being released to the ocean floor or the seafloor, but yes, definitely, we can calculate just a simple mass balance calculation. What would be, how much rock do you need to alter to basically
calculate. Of course, this is the topic of a lot of research from Dr. Arcelia's group. No, also Nicole Pelejera's research is on hyperalkaline springs in Palawan. So this is an exciting field, and ang daming analogs kasi nitong or examples ng hyperalkaline rocks and springs dito sa Pilipinas. So
definitely, yes.
That's great. So paki-compute.
Yes, try.
Okay. All right. Thanks, James.